so I hope there'll be no interference. Okay, so this afternoon, we are going to cover these two topics, Chlamydiaceae and Mycoplasma. The mycoplasmas are what you'd say minor bacterial pathogens, but are now emerging as important. Some species are emerging as important agents of non-gonococcal urethritis, especially the ureaplasma species. So we look at some aspects of that. Chlamydiaceae, an important family. It has an important pathogen called Chlamydia trachomatis that is responsible for several conditions, urogenital tract conditions, eye infections that can complicate into blindness, as well as other species that are minor pathogens but are significant causative agents of respiratory tract infections. Okay, so we look at this. You may not have a lot as far as practicals are concerned with these two organisms because of the nature of propagating them in the laboratory, but I'm going to cover some aspects of laboratory diagnosis. So we'll begin with Chlamydiaceae, and there are two important genera, Chlamydia and Chlamydophila. This is a recent classification, or a recent creation, the Chlamydophila genus. Okay, otherwise, old microbiology textbooks grouped the species under Chlamydia. So important characteristics, these bacteria are obligate intracellular microorganisms. Actually, old books, or let me say old microbiology classified chlamydia as viruses because of their nature, that when they're outside living hosts, they're metabolically inert. Okay. However, they've been reclassified into bacteria. Significantly, the infections caused by these pathogens can be treated using antibacterial, antimicrobial agents. So they're obligate intracellular microorganisms, and in clinical specimen, you can visualize them inside the host cells. In terms of their structure, they possess inner and outer membranes similar to what you'd find in the structure of a gram-negative bacteria. However, these bacteria do not stain very well with the gram stain, so we visualize them using other stains, okay, as we will see during laboratory diagnosis. They contain both DNA and RNA, and that distinguishes them from viruses. They possess prokaryotic ribosomes, and of course, they're susceptible to antibacterial, antimicrobial agents. So this group of bacteria, one important characteristic is that they have a unique replicative cycle because of the nature that they are obligate intracellular bacterial pathogens. Okay, so they exist in two forms. One form that is inside the host cell that is capable of utilizing ATP, and another form that is outside living host cells, and that form is metabolically inert. So the elementary bodies at the metabolically inert form, these are the infectious form, okay, that are resistant to many harsh environmental factors. While the reticulate bodies are the metabolically active form that are usually found in the host cells, they can multiply through binary fission, okay, inside the host cells, and they can be visualized when you stain the host cells with special stains. Okay, so in summary here, this diagram representing what happens in the replicative cycle of chlamydia. If you look at the top left here, you have the bacterial pathogen that can get into a host cell. After attachment, it gets into the host cell through endocytosis, and then it exists in its own vacuole, okay, in an endosome, which does not fuse with lysosomes. So the lytic enzymes that could be present in the lysosomes do not break or do not destroy that bacteria. So when it's inside the host cell, it reorganizes itself into reticulate bodies, which can utilize ATP from the host cell. Okay, these reticulate bodies are metabolically active. They replicate, so they can multiply by binary fission. Okay, once they have multiplied to their full capacity, they reorganize themselves into elementary bodies. And two differences in terms of species for the Chlamydia pneumonia, for the Chlamydophila pneumoniae and Chlamydia trachomatis species, they exit the host cell through reverse endocytosis. While for one species, Chlamydophila psittaci, they actually destroy the cell. They cause cell lysis, okay, and kill the cell. Now you can visualize these bacterial pathogens in host cells, for example, in clinical specimen like discharge from a patient with an STI or discharge from an eye infection. You can visualize the pathogens in these inclusion granules. So there are three medically important species: Chlamydia trachomatis, Chlamydophila pneumoniae, and Sitachi species. We'll begin with Chlamydia trachomatis, which is the most common and the most important in terms of the clinical conditions associated with it. Now, this species can be divided into several serovas based on the antigenic difference found in the major outer membrane protein. Remember, I told you it has two membranes, like a gram-negative bacteria. So the outer membrane protein, there are several antigenic differences, okay, in these proteins, and that is the basis for classification into serovas. These serovas are given alphabetical names, ABC, which primarily infect the conjunctiva. So these serovas are mainly associated with eye infections. We have serova D2K that primarily causes urogenital tract infections. Serova L, which is associated with pathogenesis in the inguinal lymph nodes. So looking at the clinical manifestations, the first one is trachoma, which is an eye infection. It is a chronic inflammatory granulomatous process of the eye surface that leads to corneal ulceration, scarring, and eventual blindness if it is not treated. So active trachoma is characterized by the presence of lymphoid follicles on the conjunctiva and intermittent shedding of the pathogen, the bacteria chlamydia. It is primarily a disease of children, and blindness can occur as a complication. Okay, so it's a chronic process, chronic inflammatory process. We have other infections by Chlamydia trachomatis in the eye, which are acute. Okay, that is called the inclusion conjunctivitis. But this chronic process is trachoma. It leads to corneal ulceration and eventually blindness. So trachoma spreads from one individual to another by spread of the discharge from the eye. And this can occur through contaminated hands, sharing clothes, sharing items like towels, okay? And houseflies also act as important mechanical vectors of transmission. When they get onto the eye or on the face of a child or an adult who has discharge in the eyes, they can move on to another individual and carry the bacterial pathogen present in the discharge. So factors that enhance spread of this disease are poverty, where you'd now have this sharing of personal effects, lack of adequate space, overcrowding, poor personal hygiene, okay, and inadequate water. This disease is more prevalent in dry, arid, and semi-arid areas, okay, even in Kenya. It's one of the diseases that are classified as the neglected tropical diseases. And it's more prevalent in these dry, semi-arid areas because of inadequate water. Water would be very, very important for personal hygiene, for washing faces, okay, to minimize chances of spread of infection from one individual to another. So this disease has been classified. I mean, the stages of trachoma disease, there are 5 stages which have been properly described by WHO. We have seen an important complication that can arise is blindness, okay? So stage 1 is where infection occurs, and this can be visualized by presence of small bumps under the eyelid after exposure of the eye to an infected discharge, okay, either from infected hands or towels or houseflies coming into contact around the eye surface, somehow the bacteria getting into the eye. And this stage 1 is represented by follicles. You can see the follicles under the eyelid. The individual will complain of feeling a sensation of small grains or some bumps under the eyelid. Stage 2 is an inflammatory process, a reaction to the presence of the pathogen. So follicles and inflammation and reddening. Stage 3 is formation of scar tissue. So in stage 1 and 2, it is easy to examine. You can see you can easily turn the eyelid inside out to examine it. And the patient comes, you can see a lot of reddening, inflammation, some bit of swelling, okay, and the follicles present. Stage 3, when the scar tissue forms, it becomes difficult, okay? After the inflammatory process, there's formation of scar tissue. The eyelid is heavy. It's difficult to turn it. It's not as malleable, okay, as a normal eyelid. It's difficult to turn it inside out for examination, okay? And as the scar tissue forms, it draws the eyelid. This tissue up here, the eyelid tissue, it draws it inwards, and that leads to in-turning of the eyelids towards the cornea. When that happens, the eyelashes cause abrasion on the cornea. There's scratching and abrasion. Okay, trachoma with trichiasis. Because of this abrasion or scratching of the eyelashes on the cornea, and it is quite painful, there is ulceration that can occur. Okay, this process of trichiasis, in-turning of the eyelids, okay, and formation of ulcers, ulceration, and abrasion makes the eye susceptible to infections, secondary infections with other bacteria like Pseudomonas, and even fungal spores that may be in the environment, okay? So, and viruses. So there's risk of secondary infection with other microorganisms. And after ulceration, stage 5 comes in where you have corneal opacity. Usually by stage 5, blindness is inevitable. It's easier to manage this disease in the early stages, okay? A second clinical manifestation is adult inclusion conjunctivitis. This is also infection in the eye, all right? Just to point out, the initial manifestation, trachoma, is due to serova A, B, and C, commonly associated with serova A, B, and C. Okay, so in adult inclusion conjunctivitis, it's an acute condition where the infected adults present with a mucopurulent discharge, dermatitis, corneal infiltrates, and in chronic disease, when it's been there for some time, there's corneal vascularization. So it is most prevalent in sexually active young people, being spread from the genitalia to the eye due to poor personal hygiene. So the serovas that are associated with adult inclusion conjunctivitis are usually serovas D to K. D, E, F, G, all the way to K. Those serovas commonly cause urogenital infections, okay, where the patients have symptoms that include discharge in the genitalia. So because of poor personal hygiene, okay, there's self-contamination, spread of that discharge from the genitalia to the eye through contaminated fingers, and that results in this inclusion conjunctivitis, okay? Then we have neonatal conjunctivitis. This condition is also caused by serovas D to K that is usually associated with genital tract infection. So what happens is the neonates get infected when they pass through the birth canal of a female who has an urogenital infection during delivery. Okay, so neonatal conjunctivitis is an acute process characterized by mucopurulent discharge, and it develops in infants a few days after birth. The infants present with swelling of the eyelids and orbit and a purulent discharge, okay? Purulent infiltration of the conjunctiva. It's acquired from the mother during birth, and quite a big number of these children, a few days after having the symptoms of neonatal conjunctivitis, they develop chlamydial pneumonia because they cry. Because of this condition, they'll be crying a lot, highly irritable, and sometimes they end up inhaling, taking in the discharge. As they cry and it runs down their cheeks, they can end up inhaling, taking in the discharge that contains the bacteria into their respiratory tract, and then they develop chlamydial pneumonia about 6 weeks after these clinical manifestations of neonatal conjunctivitis. Okay, so because of the origin of infection acquired from a mother who has an STI, the serovas associated are usually the urogenital related serovas, the D to K. So you can have C. trachomatis genital tract infections in men. As I've said, the serovas associated are usually D to K, and what they cause non-specific urethritis. The main presentation is urethritis, non-gonococcal urethritis. So the infection can be asymptomatic with the infected men serving as a reservoir of infection. For the symptomatic patients, or when they move from being asymptomatic to symptomatic, they present with mucopurulent discharge. Occasionally, this progresses to epididymitis or prostatitis. Okay, it is likely that chronic chlamydial epididymitis may eventually lead to occlusion of the tube and infertility due to azoospermia. So there's a likelihood of a complication of formation of chlamydial strictures, okay, that can lead to occlusion of the tube, and that will lead to infertility due to azoospermia. Now, genital infection can occur in women, as I have mentioned, and some women harbor this organism, Chlamydia trachomatis, asymptomatically in their cervix. So this is a risk to their sexual partners because the sexual partners can get infected. It's also a risk to their offspring in case they are pregnant. The offspring can get infected during delivery. It's also a risk to themselves because Chlamydia trachomatis frequently ascends the fallopian tube if it is not treated. So in symptomatic women, C. trachomatis cerevisiae causes mucopurulent cervicitis and urethritis. Okay. In our lecture in Syria, remember I mentioned publications state that 30 to 50% of females who have gonorrhea disease are actually co-infected with Chlamydia trachomatis. Okay, so the symptomatic individuals will present with mucopurulent cervicitis and urethritis. The infection can progress to endometritis. When you have this ascending infection, it can progress to endometritis followed by fallopian tube infection leading to acute salpingitis. That can lead to formation of scars. Okay, so collectively, endometritis and salpingitis are known as pelvic inflammatory disease, and it is largely known the world over that the most common bacterial pathogen associated with PID, pelvic inflammatory disease, is Chlamydia trachomatis. So further complications can occur. Chlamydial pelvic infection may lead to further abdominal involvement and formation of pelvic adhesions. So you can have perihepatitis and periappendicitis occurring as further complications. Another clinical manifestation of Chlamydia trachomatis is lymphogranuloma venereum, abbreviated as LGV. So this one is associated with serovirus L, subtype L1, L2, and L3. It usually begins with a genital ulcer, okay? So it's one of the bacterial pathogens that causes genital ulcer disease. So it begins with a genital ulcer followed by lymphadenopathy of the regional lymph nodes. Those are the inguinal lymph nodes. If infection persists from this point of primary infection where you have the genital ulcer, you will be able to see buboes. Okay, it can also spread to the gastrointestinal and genitourinary tract, causing strictures and in rare cases penoscrotal elephantiasis, okay, because of formation of the chlamydial strictures along the genitourinary tract. So to make a lab diagnosis of these infections or these clinical conditions of chlamydia can have three approaches. One is using microscopy, where you have microscopic examination of clinical specimen for cytoplasmic inclusion bodies, those bodies that I showed you that can be seen where replication is taking place inside the host cells. So these inclusion bodies contain a lot of glycogen. C. trachomatis inclusion bodies contain a lot of glycogen. So a simple way to visualize them is by staining with iodine. All right. You can also stain with James stain. You can use fluorescent antibody staining or hybridization. You can also use DNA probes. Okay. Another approach in lab diagnosis is serology, where you detect the antigens in clinical specimen using ELISA kits. And you can also use culture, but because these are obligate intracellular bacterial pathogens. Kindly mute. Kindly mute, please. So these are obligate bacterial pathogens. So if you want to make a lab diagnosis using cultures, you need to use living cells, the way you'd propagate viruses. So you can use chicken embryo yolk sac or the McCoy cell lines. For treatment, LGV, you can use doxycycline. You may need to aspirate the buboes if they have formed. Okay, genital and ocular infections, okay, azithromycin or doxycycline. For the newborns with conjunctivitis and a sequel of pneumonia, you can treat them with erythromycin. For control, chemoprophylaxis using oral azithromycin. public health education plays a very important role as far as hygiene is concerned. And because we have STIs here, it is important to treat infected patients as well as their sexual contacts to break that chain of transmission. Okay. Just a flyer here that was used by Jimmy Carter, the former president of US. The Carter Center was doing a lot of work in trying to reduce trachoma. And you can see here some element of hygiene. Wash your face, and of course you need to provide that water. Chlamydophila pneumoniae, the other important species for this family, Chlamydiaceae. This pathogen is spread via respiratory secretions. It's mainly associated with respiratory tract infections. It's primarily a human pathogen, no animal reservoir, so it does not cause any zoonotic illnesses. None has been documented as of now. So the clinical symptoms, the clinical conditions it's associated with include bronchitis, pneumonia, sinusitis, and atypical pneumonias. So 50% of people have serologic evidence of having been exposed to this bacteria at some point, and it's a significant cause of acute exacerbations of asthma. Diagnosis is made using serology. Okay, so ELISA kits to detect antibodies against Chlamydophila pneumoniae, PCR, nucleic acid probes, and PCR, and cell cultures, okay, of respiratory tract specimen. For treatment, macrolides are very, very effective, like erythromycin, azithromycin, clarithromycin, tetracycline, or levofloxacin. Okay, the last species of this genus Chlamydophila is Chlamydophila psittaci, and this one is associated with birds that are called the psittacine group of birds. These are the exotic birds like parrots, etc., that are usually kept as pets. However, it can also infect domestic birds. Okay, so human beings get infected when they inhale the organism that are in the droppings of the birds. As you clean the cages, if you keep these birds as pets, as you clean the cages, sweeping them, or the big houses, you could inhale the dust that has the organism from the infected bird droppings. Incubation period is about 10 days, and the clinical manifestation of infection, the disease ranges from mild influenza-like syndrome, but it can progress to more severe illness. The patient can present with general fatigue, fever, anorexia, sore throat, headache, photophobia, and it can also lead to severe illnesses that present with delirium and pneumonia. Diagnosis is mainly through serology, ELISA kits to detect antibodies in the serum, and treatment agents like tetracycline, doxycycline, erythromycin can be used. The main approach of control or prevention is proper care of these psittacine birds to treat the birds that are infected. So the vets would play an important role here, so that you treat the birds so the human beings who are handling these birds or keeping them as pets do not get exposed to the bacterial pathogens. As you take care of these birds, you need to use PPEs like masks because of the likelihood of this infection being an occupational hazard. Okay. So this table here summarizes what we have looked at as far as chlamydia and chlamydophila concerned. There's a list here on the extreme right of the organisms, the particular serovus, the site of the body where infection can occur, and the associated diseases. So it's a table summary of what we have looked at. Okay. Now the next genus we are looking at are mycoplasmas. And mycoplasmas is a term used to refer to any members of the class, the mollicutes, which include Mycoplasma and Ureaplasma genus. There are over 100 different species that have been identified. Now, this is a unique bacterium. It's the only bacterium that does not have a cell wall, all right? And it causes a wide range of infections, which can be the primary infections, as well as other infections related to autoimmune reactions to this bacterial pathogen in the body. So it's predominantly transmitted by human-to-human contact, and it primarily affects the mucosal areas of the respiratory tract and urogenital tract. So primarily, It's respiratory tract and urogenital tract, but we can see complications can arise that can affect very many other parts of the body. So I've mentioned over 100 species have been identified, but the most common species of medical significance include the most important one for Mycoplasma is Mycoplasma pneumoniae. It's the most widely studied and it's the most prevalent. So besides the respiratory tract, Mycoplasma pneumoniae has been identified in pathogenesis in other anatomical sites such as the skin, the CNS, blood, heart, and joints. And I'm going to elaborate that when we're looking at possible complications of Mycoplasma pneumoniae infections. M. genitalium and M. hominis cause genitourinary infections, and they are spread through direct sexual contact. Numerous commensal mycoplasmas also exist in the body, primarily in the mouth, the oropharynx, and the most common are Mycoplasma salivarium and Mycoplasma orale. So for the Ureaplasma genus, the most important species is Ureaplasma urealyticum, which has been identified as a cause of urologic, gynecologic, and obstetric morbidity with associated complications in men, women, and neonates. So pathophysiology of this organism, Mycoplasma has developed a specialized organelle for attachment to host cells. And this is mainly, as I've said, the mucosal cells in the respiratory tract and urogenital tract. So the attachment of Mycoplasma to a host cell prevents ciliary clearance of the organism. That's along the respiratory tract. So hydrogen peroxide and superoxide radicals produced by the Mycoplasma cause oxidative damage to the host cells. So cellular damage causes activation of the innate immune system, triggering chemotaxis and cytotoxic effects of cytokine release. So Mycoplasma can invade tissues directly and replicate intracellularly in other organs. Okay, you can also have autoimmune reactions that contribute to extrapulmonary effects of Mycoplasma infection. The primary site along the respiratory tract pathogenesis is due to production of hydrogen peroxide and superoxide radicals. Okay. So clinical manifestations, clinical presentation can vary from mild to severe illness. And multiple manifestations are dependent on site of infections, but it most commonly correlates with respiratory tract infection. The patients may present with fever, headache, sore throat, and a cough. Okay. For urogenital infections caused by Ureaplasma species, Mycoplasma genitalium, and Mycoplasma hominis, the patients can present with non-gonococcal urethritis, dysuria, that's pain while passing urine, and urethral discharge in men. Many infected women are asymptomatic but may experience pelvic pain if pelvic inflammatory disease develops. Now, I've mentioned complications that can arise, and these are autoimmune reactions, okay? And this is mainly because of some antigenic properties of Mycoplasma, okay? For example, Mycoplasma are capable of changing or altering the I antigens that are usually found on red blood cells. When that happens, you have autoantibodies produced by the host. These autoantibodies destroy the red blood cells, and that can lead to hemolytic anemia. Okay, so those are autoimmune reactions that can occur because of certain antigenic properties of Mycoplasma that can lead to changes, okay, autoantibodies being produced and certain changes. So in the notes that I'll share, I'm going to elaborate more on those antigenic properties of Mycoplasma that can lead to autoimmune reactions. One I have mentioned is the ability of the pathogen to alter the I antigens on red blood cells. Okay, so some of the complications of infection include encephalitis, cranial nerve pulses, acute disseminated encephalomyelitis, and optic neuritis. So these are complications in the CNS. Can also have dermatological complications, skin involvement, synovial fluid involvement that causes septic arthritis, cardiac complications leading to pericarditis and endocarditis, hemolytic anemia, as I have said, due to alteration of I antigens. And Ureaplasma and Mycoplasma genitalium have links to pregnancy complications, for example, preterm birth, low weight birth infants, and bronchopulmonary disease in preterm neonates. Okay, so this bacterial pathogen is very common in causing bronchopulmonary disease in neonates of low birth weight, okay, preterm neonates. Mycoplasma hominis has also been implicated in mid-trimester abortions and early miscarriages. So diagnosis of Mycoplasma infection is usually made clinically. Okay. However, one may use these techniques to confirm and support diagnosis. You can culture the pathogen in lipid-rich media. Okay, I had mentioned an important characteristic is that Mycoplasma does not have a cell wall. Another important biological characteristic is that it contains cholesterol in its cell membrane. So to propagate it in the laboratory or to culture it successfully, you need to potentiate your media with lipids. Okay, so culture it in lipid-rich media, but you see it takes as much as 8 weeks to grow. So this would not be a good approach for patient management, but it can be used if you want to isolate the bacteria for further analysis. There are serological tests based on antibody tests that can detect antibodies against the bacteria. And radiological imaging plays an important role, especially for the respiratory tract infections. So for treatment and management of conditions caused by Mycoplasma and ureaplasma, remember, because Mycoplasma lacks a cell wall, it is naturally resistant to any antibiotic that targets the cell wall, and there are quite a number. Okay, so choices for antimicrobial therapy are limited to those that do not target the cell wall. Macrolides, tetracyclines, and fluoroquinolones have been found to have the highest efficacy against Mycoplasma infections. Doxycycline is the first choice of treatment for M. hominis and ureaplasma urolithecum related infections. Fluoroquinolones are useful for immunocompromised patients and unusual cases of systemic infections when you have the complications that I have mentioned. So the length of treatment will vary depending on the patient's condition and the extent of illness. Okay, so as of other STIs, it's important that sexual partners of patients who have STI infections, it's important that the sexual partners are also treated to break the chain of transmission. Okay, so that is our discussion on chlamydia and mycoplasma. I say they are minor pathogens, but I want to emphasize that ureaplasma urolithecum is now emerging as an important causative agent of non-gonococcal urethritis, especially in men. There are some publications that were released recently, I saw from Nigeria, that pointed out to the increased prevalence. Okay. Any questions? So if there are no questions, I'll release you so that you make your way to Kenyatta, for those who are not in Kenyatta, for your physical practicals from 2 p.m. I will share the notes with your class reps or through your class email. Kindly read them. Okay. Have a good afternoon. Thank you.